today we're going to talk about Chapter 5, and Chapter 5 deals with eukaryotes. Okay? In Chapter 4 we talk about prokaryotes, primarily bacteria. Now we're going to expand upon that a little bit and talk about eukaryotes, or cells that are a little bit more organized, a little bit more higher order in nature. So, we're going to focus on these guys today, the eukaryotes. This is a very diverse group of organisms. Okay? And here we're primarily going to be talking about fungi, protozoans, and helminths. Okay? Those are the three groups that fit in within the microbiology realm. Okay? But keep in mind that there are many, many other types of organisms in the eukaryotic kingdom. A couple examples would be humans. Okay? We are eukaryotes. So just because we're talking about only these three groups, don't get in your head that that's only what eukaryotes are. Okay? Now, as we mentioned before, these guys are more complex and they can be much larger than prokaryotes. They can also form large multicellular organisms like ourselves or other animals. Okay? So it is important that we talk about eukaryotes because they can be pathogens. And in fact, when they are pathogenic, they tend to be much more difficult to treat or eradicate because they are very similar to ourselves, okay, when you really break it down. So it's important that we talk about them, and we'll bring up some good examples of some parasitic or pathogenic varieties of eukaryotes as we go. And then in Chapter 6, we'll talk about viruses. And again, these are not really alive. They're just little particles that can infect cells and take them over. So a little bit about the history of eukaryotes. We briefly talked about this in Chapter 1. But eukaryotes haven't been around for that long, at least compared to prokaryotes. They've been on Earth for about 2 billion years. Okay, so about two-thirds or a third of the Earth's lifespan. Now, it is thought that eukaryotes actually formed from a bunch of prokaryotes that kind of stuck together and decided that it was better to live together than by themselves. Okay? And there's some reasons why we believe this. If you look at certain organelles within eukaryotes, they actually look a lot like prokaryotes. Okay? A prime example would be the mitochondria. So if you look at the mitochondria, it actually looks a lot like E. coli. It has its own circular genome, and it can reproduce within the eukaryotic cell just like a bacteria would. Okay? So there are reasons why we believe this. It's not something we just kind of made up. We think that this occurred because it was better for this new eukaryotic organism to live with a bunch of prokaryotes inside of it, because it allowed for the production of what we call organelles. And these organelles are structures within eukaryotes that allow for compartmentalization of functions. What that means is now eukaryotes can streamline these functions, make them extremely efficient, and thus become larger and more complex. Okay? So this building of a eukaryotic organism from multiple prokaryotes allowed for the complexity that eventually became us. Okay? So there are some notable differences between eukaryotes and prokaryotes, and these are things that we're going to bring up throughout the semester. Okay? And as I mentioned before, it's important to understand the difference between prokaryotes and eukaryotes, because prokaryotes, aka bacteria, are the ones that are often causing disease in humans. So by understanding these differences, we can particularly target the prokaryotes while leaving the eukaryotes ourselves and other animals alone. So some of the big differences, first and foremost, is they are larger in size, which we've already talked about. Usually about 30 times larger, okay, than most prokaryotes. So they're significantly bigger. They contain a nucleus. And a nucleus is basically a little cell within a cell, and this is where the DNA is stored. Okay? So remember, in prokaryotes, the DNA is just floating around, kind of willy-nilly in the cytoplasm. In eukaryotes, it's actually found within the nucleus and only in the nucleus. And then we have organelles. And as I mentioned before, organelles are structures within the eukaryote. You can see them here as all these colorful little structures or blobs. And their primary purpose is to do very specific functions extremely well. Okay? And by doing this, it allows the cell to streamline processes, making things more efficient, and making the cell larger and more complex. So as we mentioned, in microbiology terms, we're primarily going to focus on protozoans, fungi, some algae, but not really, because they're not pathogenic, and helminths. So the big three guys here are the protozoans, the fungi, and the helminths. These are all eukaryotic organisms, and they can all be pathogenic. Okay? Protozoans are single-celled animal-like cells. Okay? Most of these are free-living, but some can cause disease. Fungi are kind of in their own little subkingdom, and they are what we call terrestrial saprophytic organisms. And most of the time, they are going to break down dead, decaying material. However, there are some that can cause disease in humans, and those are the ones we'll focus on. And then multicellular organisms, the only ones we're going to focus on in the microbiology side of things are going to be the helminths. And these are larger worm-shaped organisms. Again, most of the time they will be parasitic. So we'll bring up some specific examples as we go. So this is a nice little cartoon diagram that shows all the bits and pieces of a generic eukaryotic cell. Okay? This is not a real cell. You would never see all of these structures in one cell. Okay? However, this gives you a good idea of what all of these bits and pieces look like and where you would find them. So we'll start off in the center, this big purple blob here, and this is known as the nucleus. As we mentioned before, the nucleus is found in the center of the cell, and it is primarily there to protect and store the DNA, okay, or the chromosomes. This is different than prokaryotes, because remember, prokaryotes have their chromosome just floating around willy-nilly. The nucleus itself is covered by what we call a nuclear envelope, and this is a double plasmid membrane, so kind of like a cell membrane, but twice as thick. And it has little holes in it known as nuclear pores, and this is the only way things can come in and out of the nucleus to interact with DNA. So the main purpose of having this nucleus is to protect the DNA itself. Only when the cell lets things in these little nuclear pores can it come into contact directly with the DNA itself. And conversely, only when things are let out of these pores are they allowed to interact with the rest of the cell. So it adds a layer of protection and control, again, required in eukaryotes because they are larger and more complex. Around that, we have the endoplasmic reticulum. More specifically, this is what we call the rough endoplasmic reticulum. We call it rough because if you look real close, you can see all these little dots that kind of stick all around that part of the ER. The rough endoplasmic reticulum is responsible for protein production, and those little dots that you see are ribosomes. Remember, in prokaryotes, ribosomes are used for the production of protein. The same thing applies for eukaryotes. However, instead of just pooping the protein into the cytoplasm, it poops the protein into the endoplasmic reticulum, where it then gets processed, folded, and turned into an actual protein. Next to that, we've got these little smokestack-looking things. This is what we call the smooth endoplasmic reticulum, or the smooth ER. And this is used for one of two things. Detox reactions, removing toxic substances from the cell, as well as lipid metabolism, or processing of lipids and oils, which sounds kind of stupid and easy, but it's actually really complex when you think about it. Below that, this little blue guy is the mitochondria. This is the guy that looks a lot like E. coli. It has its own DNA, its own chromosome, and it can reproduce on its own. Everything else in the cell, other than this and chloroplasts, have to reproduce only when the cell is divided. The mitochondria is primarily used by the cell to produce ATP, which is an energy store, and it can be used by the cell to make things or do things. Next to that, we have the chloroplast. The chloroplast is found in autotrophs and photosynthetic organisms. And this is primarily used by the cell to produce glucose. So this is something you would find in plants, not so much in animals or fungi. Okay? Its primary purpose is to make glucose, which the cell can then store and use later, or if that cell gets eaten, it can be taken up by other organisms and used as an energy source. Right above that, you have the Golgi apparatus, a bunch of flat pancake-looking things. And the Golgi apparatus is what allows the cell to be as large as it is. The Golgi apparatus is used for transport. So it picks things up and moves it around the cell. So Remember in chapter 4 I said that prokaryotes can't get that big because they're limited by the volume that they have? Well, eukaryotes aren't limited that much by volume because of the Golgi, because it can move things around very easily. Down here we have centrioles, which are used for mitosis or division, which we'll talk about briefly in a second. And that's pretty much it for the internal stuff. So what you're noticing here is that with eukaryotes, there's a lot more bits and pieces on the inside. But the good news is there's a lot less stuff on the outside. Okay? So really, when you look at the cell on the cellular envelope, there's usually either one or two layers. Okay? If we're talking about animal cells, there's just one layer, and that is the cell membrane. That's it. They have nothing else. Okay? If we're talking about plants or fungi, then we have an additional layer, and that is the cell wall. Okay? But there's no outer membrane. And sometimes there can be a glycocalyx, but not always. And you'll note that there is a flagellum in this case. However, the flagellum in eukaryotes is significantly different than prokaryotes. It's still used for movement. However, instead of spinning like a motor, it wags like a tail. Okay? But it still has the same purpose of movement. So a lot of these structures we're going to talk about are similar in nature to the prokaryotic structures. There are some little differences, which I do expect you to know, though, as we go. Okay? So let's start off with the flagellum. Okay? Note that instead of saying just flagella, we usually say flagellum, and that's because there's usually only one. Okay? Where prokaryotes, you can have hundreds. In eukaryotes, there's usually only one. As I mentioned, these do not spin, so it's not a motor. Okay? It does not move in the same fashion as a prokaryote. Instead, it wags like a tail. Okay? The way that it works is you have a bunch of what we call microtubules. These are long polymers of protein, and these form these long chains. If you take 9 of these, you can make a hollow tube with 2 in the middle. And what the cell does is it literally just yanks on these 2 like this, and this causes the tail to flow back and forth. Okay? So big difference here is the wagging instead of spinning. But still serves the same purpose. It still allows for movement, it still allows for chemotaxis and all that other stuff. Now there's an additional structure that can be found on eukaryotes externally that can be used for movement or locomotion. And that is something called cilia. So as I said before, flagella, there's usually only one, so one single flagellum. So what the organism does instead to allow for better movement as well as more motor skills is it will also incorporate these cilia. And all cilia are really small, short flagella that basically cover the organism entirely. It can move these in unison, and this allows the organism to propel forward, or it can move one side more so than the other, and this allows it to steer. Okay? It's also important to note that cilia are found not only on single-celled organisms that use them to move, they can also be found on multicellular organisms like ourselves, and we can use this to propel things through the body. So an example of where we have cilia is lining our esophagus. The cilia is there to push the mucus up so it doesn't get stuck in the lungs. We then cough and spit it out or swallow it in the chest. So these can be found both on single-celled and multi-celled organisms, and that determines their function. The glycocalyx, so just like prokaryotes, eukaryotes can have a glycocalyx, and it serves pretty much the exact same function. In fact, it's actually made out of the same stuff, polysaccharides. It's there to create a slimy covering that coats the organism. This protects it from the environment, but also protects it from being gobbled up or phagocytosed. It can be found in 2 arrangements, so slime layer or capsule. Remember, slime layer is loosely held on, or capsule is tightly held on. The functions of the glycocalyx, virtually the same as we saw in prokaryotes. It protects, it also makes them sticky so they can stick to each other and form biofilms if they want to. However, there's one additional thing that the glycocalyx can do for eukaryotes, and that is it can receive signals. So it can act as kind of like a sticky glob of stuff. It can pick things up from the environment, and this can then radiate through the cell and tell the cell what it's encountering. Okay, so it acts like a microphone, I guess, for signals. And as we talked about before, fungi and algae also have one extra layer, and that is a cell wall, where protozoans and animals do not. Okay, so fungi and algae have cell walls, protozoans and animal cells do not. Now, the capsule is very important for pathogenic organisms, especially pathogenic eukaryotes. And as we mentioned before, that's because it prevents phagocytosis. So phagocytosis is the process in which our cells in our body gobble things up that are foreign or bad. And if you have a slimy, slippery coating covering you, this becomes more difficult to do. This allows the pathogen to survive in the body and then cause subsequent disease. And as I said before, it is very, very similar to prokaryotes. The cell wall, so as we mentioned earlier, both plants and fungal organisms have cell walls. The cell wall is thick and rigid and gives external structure, just like we saw in prokaryotes. It's a similar function. However, what it is made out of is very different. Okay? In fungal cells, it is often made out of a molecule called chitin. Okay? And chitin is a molecule that is found only in fungi. It's not found in plants, it's not found in animals or protozoans. It is only found in fungi. In algae cells or plant cells in general, most of the time the cell wall is going to be made out of cellulose. Okay? Cellulose is an insoluble fiber, and that's what makes us poop, basically. And that's why we eat a lot of vegetables, to get the fiber. It's basically the cell wall from the cells in that plant. The cytoplasmic membrane, or the plasma membrane, very, very similar to prokaryotes. It's a phospholipid bilayer. It is designed to let things in and out of the cell as the cell sees fit. However, there's one notable difference with the cytoplasmic membrane in eukaryotes compared to prokaryotes. The biggest one is containing sterols. Okay? Sterols are special lipid molecules. They give extra structure to the cell membrane. This is why animal cells and protozoans do not need to have a cell wall, because they have these sterols. Now, the sterols do differ depending on the eukaryote. So in animal cells like ourselves, the primary sterol is cholesterol. Okay? In fungal cells, the primary sterol is eugesterol. Okay? It's important that I bring that up because, again, that is something found only in fungal cells. It's not found in other animals or found in plants or anything like that. All right, so let's move on to the internal structures. We'll start off with the nucleus. The nucleus, by far and large, is the largest organelle within the cell. And that's because it has all of the DNA within it. Okay? There is no DNA outside of the nucleus, at least in a healthy cell. Okay? As I mentioned before, it is made up of a double lipid bilayer. This double lipid bilayer is called the cellular envelope. And it is dotted with little holes, or what we call nuclear pores. And this is what lets things in and out of the nucleus as the cell sees fit. Again, this is important because it protects the DNA for the eukaryote. And by protecting it, the eukaryote can live for longer periods of time. So instead of living 15 minutes like a bacteria would, these cells can live for hundreds of years. The nucleolus, this is what we consider a really dense portion of DNA found within the cell itself. And this is where we make a particular type of RNA called rRNA. We'll talk more about what rRNA is in Chapter 9. The nuclear pores, again, let things in and out, and this is, gives the cell great control over what interacts with the DNA within the cell. This is an example of what a nucleus looks like in real life. This is an electron micrograph. You can see the nuclear envelope here. You can see the two layers in the nuclear envelope. And you can actually see little pores right here, these little like holes within the nuclear envelope itself. In the center, you see the nucleolus. This is the dense part of DNA. But all this cloudy stuff right here around it is also DNA. It's just not as dense. Remember that the nucleus is chock full of DNA. It's just all DNA inside of there and RNA. So a little bit about the genetic material in eukaryotes. So we mentioned the DNA is found within the nucleus. And instead of it being circular, like we said in prokaryotes, it is now going to be linear. Okay, so it comes in long strings. It is often broken up into multiple pieces called chromosomes. So instead of having just one single circular chromosome like we saw in prokaryotes, eukaryotes have multiple linear chromosomes. Now the amount of chromosomes that are present depend on the species that you're dealing with. Okay? And don't get it in your head that the more chromosomes, the higher order, the better the organism. There are some yeast that have 200, 300 chromosomes. We only have 46. Okay? So the number of chromosomes really doesn't matter much. Instead, it's the amount of material that's there. So you can have 200 chromosomes and only have 100,000 nucleotides, where we have 46 and have 4 billion. And that's what makes us more complex. These organisms can be haploid or diploid. Haploid means you have one set of chromosomes. Okay? Diploid means you have 2 sets. If you're haploid, you are considered asexual. That means you just divide without actually having to interact with cells and mate. Diploid usually means you are sexual. And so you get one chromosome from mom and one chromosome from dad, and the cell just randomly chooses which one it wants to use at that time. The DNA itself is very, very large in eukaryotes. Again, the DNA is bigger because you have to do a lot more stuff when you're a eukaryote. And so a lot of times the DNA is wrapped around proteins called histones, and this allows it to condense down. If it didn't do this, it would be so big, it would be much larger than the cell itself. So it wraps around protein, creating kind of big balls of yarn of DNA, and this keeps it small and condensed. Now, eukaryotic cells go through division, and depending on the type of cell, they can either go through mitosis or meiosis. Mitosis is where you are dividing your normal cell, you're going from one cell to 2. Meiosis is what occurs when you want to produce gametes. These are haploid cells from a diploid cell, and these are used for mating. So most cells are going to go through mitosis. Let's jump back. Okay. So, what is mitosis? I'm not going to go into too much detail on this because we'll talk about it in anatomy and physiology when we get there if you haven't already. But mitosis is a little bit more complicated than what we see in prokaryotes. There are many steps involved, and that's because the cell has to duplicate all of it, not only its DNA, but also all of its organelles and things before 2 cells can be formed. So up at the top we have what we call interphase. And this is where the cell spends 95% of its life. This is where it is just doing its daily routine, right? It's eating, it's growing, it's doing whatever. It's not actually dividing at this step. Then something triggers in the cell saying, hey, you have to divide, and it starts to go into mitosis. The first step is you go into prophase, and this is where the chromosomes will actually duplicate themselves. And this is really important because you want one set of chromosomes for each new daughter cell. Also at this step, those centrioles that we talked about before will go to either end or either pole of the cell, and they create these spindle fibers which allow for the splitting of the chromosomes in the next step. The next step, metaphase, this is where the nuclear envelope dissolves and the chromosomes are attached to these spindle fibers, and then they are literally pulled apart in the next phase known as anaphase. This separates the 2 sets of chromosomes, one on either end of the cell, and eventually this will allow for telophase, which causes the cell to constrict in the middle and form 2 new daughter cells. Okay? So there's a couple steps involved, but again, I don't expect you to know too much detail about this other than it's complicated. The endoplasmic reticulum, as we talked about before, there are 2 types, the smooth and the rough. The smooth is used for lipid metabolism and detox reactions, where the rough is primarily used for protein synthesis. And remember, it is called rough because it has ribosomes stuck to it. So here you can see an example of the rough. You see all these little dots, and if you zoom in, those are ribosomes. And those ribosomes literally just sit there producing amino acid chains and pooping them into the endoplasmic reticulum. The ribosome itself, so we talked a little bit about ribosomes in chapter 4. They are there to produce protein. Okay? This is the site of what we call translation, and this is where RNA is translated into amino acids and eventual proteins. The eukaryotic ribosome is made up of 2 units, the large subunit and the small subunit, so much like we saw in prokaryotes. However, the large subunit is much larger, and so this results in a larger size. Also, as I've talked about before, the eukaryotic ribosome is significantly different in the way that it goes about making protein than compared to prokaryotes. And so this allows us to do something called selective toxicity, which means we can target a specific type of ribosome, let's say a prokaryotic ribosome, disrupt it and destroy it, while leaving our ribosomes alone. Okay? And this is essentially how a lot of antibiotics work. You target the prokaryotic ribosome, killing the cell, but leaving yours alone because of these significant differences. Mitochondria. So mitochondria are what we call the powerhouse of the cell, and its primary purpose is to make ATP. This is the site of cellular respiration, and as you guys will learn, ATP is used by the cell to do anything that requires energy. It is essentially a short-term energy store, and so it allows the cell to function. Without ATP, it quickly dies. The mitochondria itself looks a lot like a gram-negative bacteria, much like, let's say, E. coli. And we say that because it has an outer membrane, an inner membrane, and it has its own circular chromosome, much like a bacteria. It does reproduce on its own, and again, we think it is because this is a bacterial endosymbiont. It has lost its cell wall, though, so it does not have a cell wall. It is just 2 layers, the outer and inner. Chloroplasts. The chloroplasts, again, are the site of photosynthesis, which means they are found only in photosynthetic plants. It is thought to also be a bacterial endosymbiont, again, because it has 2 layers, an outer and inner membrane, and its own circular chromosome. It also reproduces on its own, just like the mitochondria does. And the essential role of the chloroplast is to make glucose. Glucose is a complex carbon molecule that can be produced from inorganic compounds like water and carbon dioxide. This is essential for life because this is how we get the building blocks of all organic molecules. The Golgi apparatus. In the Golgi apparatus, this is that stacked pancake-like structure, and it is primarily used for trafficking, or moving things around the cell. It sounds kind of meaningless, but in reality it's actually really important because without this, the eukaryotic cell would be constricted in size. It wouldn't be able to be as large as it is. It serves important functions, not only moving things around inside of the cell, but it can also secrete things from the cell fairly easily. The cytoskeleton. So just like in prokaryotes, there's also internal scaffolding or structures that can keep the cell together. These are seen as actin, microtubules, intermediate filaments, or microfilaments. Essentially, these are just long polymers or chains of molecules, and the primary purpose is to give internal structure. These become really, really important in some eukaryotes because a lot of times, especially in animal cells, you do not have a cell wall. So without a cell wall, you don't have that external structure, so instead you need to rely only on your internal structure, and that is the cytoskeleton. Vacuoles. So vacuoles are basically holes within the cytoplasm where things get stored. Okay? We talked about inclusion bodies with prokaryotes. Vacuoles are very similar. Okay? They store things. They can store things like glucose for rainy days, for energy stores. But also, our cells can store things like lysosome, which is basically a bunch of enzymes and things that it can be used to break down material, kind of like a digestive tract. So really, this is just a place to store material for a later day. Now, where do lysosomes come into play? Well, we mentioned earlier, phagocytes. These are cells that are designed to gobble up and get rid of any foreign things in the body. And the way that phagocytes do this is they will engulf whatever they want to destroy, and then they fuse that with a lysosome. The lysosome carries around nasty enzymes and reactive oxygen species, which tear up whatever is found within that phagocyte, destroying it and ridding it from the body. So these are very important in humans as well, because they can help clear pathogen during infection. Okay, so those are all the little bits and pieces of a eukaryotic cell. Now what we're going to do is talk about some kingdoms within eukaryotic. Okay? There are three that we're going to focus on: fungi, protozoans, and helminths. Okay? We'll briefly talk about algae, but algae is not pathogenic, so we'll just skip over most of it. So we'll start off with the kingdom of fungi. And this is a very large kingdom. There are over 100,000 different species of fungi. And these are very complex, and they are always terrestrial, which means we only find them on land. They do not live in water. One distinct characteristic of a fungal organism is the ability to produce something called hyphae or pseudohyphae. And hyphae, as you can see here, are just organized structures that the fungal cells produce. Okay? When we say hyphae, this usually means a multicellular fungal organism. And this is where you have multiple fungal cells kind of stacked together in a row, forming these hair-like structures. Pseudohyphae are what form in single-celled fungal organisms called yeast. And these are just little blobs that kind of hang off the end of the cell. Okay? But they are very distinct for fungal organisms, and it's one easy way to tell if you're dealing with a fungus organism. And so, as we mentioned, they can be unicellular, so single-celled, or colonial or multicellular. It just depends on the organism. In fact, some fungal organisms can be unicellular one day and multicellular the next. Okay? This allows them to do things like cause disease in humans. So they are terrestrial, they are not photosynthetic, and oftentimes they are saprophytic. Saprophytic just means they like dead, decaying material. So most fungal organisms are going to break down dead stuff. Okay? So most of the time, fungal organisms are not going to be harmful to humans, because their main purpose is to go after dead things, not living things like ourselves. Single-celled fungal organisms are known as yeasts, and multicellular fungal organisms are known as molds. As I mentioned, distinguishing characteristic of fungal organisms is hyphae, and this is just a series of cells kind of stacked together in a string. And when you have a bunch of these hyphae next to each other, this becomes what we call the mycelium. Earlier we mentioned that in the membrane they have a special sterol called eugesterol, and in their cell wall they have chitin. These are two molecules that are unique only to fungal organisms. And I keep bringing this up because it's important. If you get infected with a fungal organism, you need a way of destroying it. And the way that you do that is you target differences in the fungal organism compared to ourselves. And the two biggest differences, eugesterol and chitin. So it's important that we understand these differences so that we can target these cells if needed. It can be either haploid or diploid, and that means they can be either sexual or asexual. It just depends on the movements. So here is an example of a mold, both on some raspberries and also on someone's foot. Okay? So they can really grow all over the place. But as I mentioned, most of these are going to be saprophytic and like dead decaying material, like these dead raspberries. But occasionally, if they get into, if you get a pathogenic variety, they can grow on the skin and sometimes in the body and cause disease. Okay? But most of the time, if you are a healthy individual, fungal diseases are very mild and easily treatable. If you are immunocompromised, your immune system's weakened for whatever reason, then they can become more serious. But most of the time, fungal diseases are mild. This is someone that is immunocompromised. This is an example of athlete's feet, right? But I don't know about you, but I've never had athlete's feet, right? Normally it gets a little red, burns, and that kind of thing. It doesn't start like peeling off and blackening your skin. So, how are these guys organized? Okay? As we mentioned, if they are single-celled fungal organisms, we call them yeast. Okay? And if they are multi-celled organisms, or fungal organisms, we call them molds. These molds can form into cottony, hairy, velvety textures, and this is essentially all of those hyphae that we talked about, those hair-like structures, kind of growing out all over wherever the organism is found. When you have a lot of hyphae together, this is known as the mycelium, and this mycelium can be used for one of two purposes. It can be used to take up nutrients, but it can also be used for reproduction. So how does this look like in a normal fungal growth cycle? Okay? So what happens initially is you get a spore, which is basically a baby fungus. And this is just a little airborne particle. They're very, very small and very light, and they are ubiquitous. So that means they are literally everywhere. We breathe them in on a regular basis, and they literally just fly all over the place. Okay? So eventually, these guys are flying all over the place so that they can land on some nutrient-rich material. In that case, it is this dead strawberry. Okay? Once the spore lands on the nutrient-rich material, it starts to grow and divide, and it becomes what we call a germ tube. And this is essentially just a little baby hyphae. Eventually, this will start to grow and branch out, and you form these individual hyphae. And they get larger and larger and larger until you form the mycelium. All the mycelium is a bunch of hyphae stuck together in a network. Now, once you have these myceliums formed, some of them will go into the material where the nutrients is, and these are what we call the submerged hyphae, or the vegetative hyphae. Their purpose is to take up nutrients, much like a root of a tree, and bring it up into the rest of the organism. The surface hyphae, this is the hyphae that is expressed on the surface, and this is the part that you actually see. Okay, this is the hairy, fluffy part. Its primary purpose is to be reproductive. The surface hyphae will eventually form these little organospores, which will rupture, release more spores, and that's how it continues the life cycle. The key here is that what you see on the top is not all that's there. So if you have moldy food, don't just scrape off the top and eat it. I can guarantee you there's a lot more below it. Okay? An example of where this can actually be beneficial is the making of some cheeses. So brie starts off as a very hard cheese, much like Parmesan. The way that they get it to be very soft and gooey is they coat it with spores from a particular fungus. This fungus then grows, submerges its hyphae in there, releases enzymes, and digests the cheese until it turns into gooey. So it can be gross, but it can also be beneficial. Okay? Keep in mind, most of the fungi that do this stuff aren't going to be the ones that cause disease. So the hyphae themselves are these long, kind of string-like structures, and they're made up of multiple cells. And these cells are separated by something called a septa. And all that is just a little wall that separates one cell from another. The reason why we talk about them is because you can actually look under the microscope and you can say, here's one hyphae, and it has this many septa. And that can actually tell you what the species of the organism is. So it's important that you know these so that when you look, you can say, okay, I know what this is or that is or this, based on some of these distinctive characteristics. So we mentioned before, the vegetative hyphae, this is the stuff that's in the actual material, and it's used to break down the nutrients and absorb it into the rest of the fungus. And then the reproductive hyphae, this is the hyphae on the surface, which are there to eventually produce spores. Now, yeasts are a little bit different. Because they are single-celled, they do not form these very organized hyphal structures. And the reason for that is because hyphae are made up of multiple cells. These guys are single-celled, so that's kind of not possible. So instead, they form what we call pseudohyphae. And these are a lot like bacterial arrangements. So when the cell divides, instead of completely separating itself from each cell, sometimes they get stuck together. And when these form chains, this is your pseudohyphae. Okay. So a little bit more about yeasts. So yeasts are single-celled fungal organisms. And we talk more about these guys than anything else because these are often the ones that are pathogenic. Why? Well, if you think about it, it's much easier for a single-celled yeast to go around the body than some big fluffy mold, right? So being a single-celled is advantageous for fungal organisms that want to cause disease. And so for this reason, some fungal organisms can be a mold sometimes and a yeast at other times. We call this dimorphic, and usually this is determined based on the temperature. So if the organism is outside of the body at room temperature, it will grow into a fluffy mold, because it's easier to spread around as a fluffy mold in the environment. But as soon as you breathe it in or get it into a cut, and it's at 37 degrees or body temperature, it stays a single-celled yeast, which allows it to get around the body much easier. Now, yeast can be useful. So an example of a useful yeast are Saccharomyces. Saccharomyces is the yeast used for making bread, beer, wines, and cheeses and things. Okay? Really useful yeast, we use it all the time. But some yeast can be pathogenic. So Cryptococcus can cause diarrhea, dysentery diarrhea or bloody diarrhea. And Candida, down here, can cause the causative organism for yeast infections, both in the vagina as well as in the mouth, or what we call thrush. So there are pathogenic varieties, but again, most of the time these are going to be on the milder side. Okay? They're not going to be that serious unless, you're immunocompromised. So how do these organisms reproduce? Well, first and foremost, they usually produce spores. Spores are just little baby cells, okay, that are very light and easily can be dispersed throughout the air. The reason for producing these spores is so that they can multiply, survive, produce genetic variation, especially if there is sexual reproduction involved, and dissemination, or spreading of the organism. Now, there are different flavors of spores. So there are what we consider traditional spores. These come from organospores. And these are essentially the little baby ones we saw in the previous picture. The little pieces of liquid dust, and they get released from the cell, from the sporogonium, and then they float around and land on things and reproduce. The key here is that they are released all at the same time. So organospores are released at once. Another flavor of spores are what we call cnidiospores, also known as cnidia. And these are spores that are released one by one via a process called budding. So organospores released at once, cnidiospores released one by one. So what do these look like? Up here we have the sporogonium and organospores in the middle. These literally rupture at once and just released at one time. Where cnidiospores over here are released one by one in a budding process. Released all at one time. Now, fungal cells can be sexual, and the way that they are sexual is by using sexual spores. The way that this works is you have two types of spores, an A and an alpha, and the only way that the fungal organism can grow into an adult is if an A lands by an alpha and they mate. Okay? So it's kind of a random process, but this allows for genetic diversity. How do we identify fungal organisms? Well, like I said before, a lot of times you can just do it under the microscope. Look under the microscope and you see this interesting hyphae that has this many septa, and from there you can come up with a diagnosis sometimes. Also, growing them, culturing them can tell you a lot as well. Unlike bacteria, these guys have very distinct colorings and very distinct growth patterns. So you can look at them and be like, oh, it's black, that means it's this. Or, oh, it's green, that means it's this. Now, fungal organisms in nature, as we mentioned before, are mostly free-living, which means they do not parasitize organisms. They do not live off of living organisms. Instead, their primary role is to break down dead, decaying material. Okay? However, there are some varieties of fungal organisms that have become pathogenic. Okay? And when that happens, then they can cause disease in humans. However, most of the time, this is only going to occur, at least in a serious manner, in people who have slightly weakened or compromised immune systems. If you have a healthy immune system, most of the time, fungal diseases aren't going to be that much of a problem. When do you have a compromised immune system? Well, you can have one if you are busy fighting another infection. So if your body's fighting something else, it's preoccupied, and then the fungal organism can come in as what we call a secondary pathogen and start to cause disease. Or it can act as an opportunistic pathogen, and that is where it only is pathogenic in people who have lost their immune system. So if you have HIV/AIDS, you have cancer, diabetes, something along those lines, where your immune system is weakened to the point where it can get in and cause problems. But it's also important to note that even if a fungal organism is not pathogenic, it still can cause problems for humans. Fungal organisms are important for allergies. A lot of them will trigger allergy symptoms, as well as poisonings, because they can release poisons into food items, and agricultural damage, which can cause lots of money. But it is important to keep in mind that fungal organisms are beneficial, or at least can be beneficial. Okay? We can use them, as we said before, to break down dead decaying material. But we can also use them in industry to make a whole bunch of stuff. So a lot of antibiotics, at least natural antibiotics, originated from fungal organisms. The first antibiotic, penicillin, came from the penicillium mold, which is a common green bread mold. Okay? So they are important in helping fight other infections, especially bacterial infections. We can use them to make alcohol, such as in beer and wine. We can use them to make organic acids, vitamins, and even food flavorings. Barbecue potato chips. One of the main flavoring ingredients of barbecue potato chips is lyophilized yeast. These are freeze-dried yeast. Freeze-dried yeast has a very what we call umami taste, which is kind of rich, kind of like a steak kind of flavor. And so if you add that to chips, it gives it a good barbecue flavor. Okay? So they are beneficial in many ways. So I don't want you to walk away from this thinking that they're all bad. But the ones that are bad can be a pain in the ass. So, again, fungal organisms that can cause disease can be problematic, but usually they're only found in people who are immunocompromised. Most of these are going to be superficial, which means they are found on the skin, okay, not deep into the body. Some of the more common ones are going to be what we call tinea, which is really a fungal infection on the skin, and it can vary, from areas in the groin called jock itch, or maybe on the feet, athlete's feet. But it can occasionally get in deeper into the body, and this is where it can cause lung and even septic infections. Again, these are relatively rare, primarily in immunocompromised people, but when they do happen, they can be life-threatening. Okay? So superficial fungal infections, not a big deal. Deep ones in the lungs and the blood, very serious conditions. Alright, so now we're going to move away from the fungal organisms and talk about the protists. Protists are essentially single-celled organisms, okay? And they can be either algal organisms or protozoans. Algal organisms are single-celled plants. Protozoans are single-celled animal-like organisms. So we'll start off with the algal organisms. Algal organisms are photosynthetic, which means they produce their own glucose and subsequent energy production from sunlight. So what this means is they are not pathogenic. Okay? Because they do not need to take any nutrients from other sources like a human, we do not have to worry about them as pathogens. So for that reason, we're not going to spend much time on them. We do classify them based on their color or their pigment. And so you have brown, red, green, euglenoid, or are blue-green algae and dinoflagellates. Generally speaking, they are important environmentally, because this is where most of our oxygen comes from that we breathe, but not so much as far as their clinical role is concerned. So instead, we're going to focus primarily on the protozoans. The protozoans are single-celled animal-like cells. Okay? And by far and large, most of the 65,000, are going to be free-living, freshwater dwelling organisms. Okay? These guys are going to swim around in fresh water eating bacteria. They're not going to cause disease in humans, except for a few exceptions. Okay? There are about 30 protozoan diseases that target humans and cause disease. So protozoans, these guys are eukaryotes, as we talked about. That's what this whole chapter is about. They are single-celled. They can be free-living or pathogenic. And we usually group them based on their locomotion. So how do they move? Do they roll around? Do they use cilia, flagella, or do they not move at all? And they typically have a life cycle that has one of two forms, trophozoite or cyst. Trophozoite is where the organism is alive, metabolically active and doing its thing. Cyst is where it kind of shuts down, becomes not metabolically active, and waits for better days. So this is kind of like a vegetative state when we talked about endospore formation, and then this would be like this endospore. So as I mentioned, these are single-celled organisms. They contain most eukaryotic organelles except chloroplasts, and that's because they are not plants. They have kind of a weird composition. They have this ectoplasm and endoplasm. The ectoplasm is a thinner layer kind of that covers the organism, and it is used to kind of undulate and move the organism around. Okay? It's not found on all protozoans, but it is unique to protozoans. They do not have a cell wall, again, because they are animal-like in nature. And they can become pretty large sometimes. So earlier on in chapter 1, we talked about all the different sizes of microorganisms. And I showed you that one that would be about the size of 10 city blocks. That was an amoeba. That is a protist. Okay? So they can get large in size, sometimes so large that you can see them very easily. Okay? As I mentioned, they have two stages in their life cycle. They have a trophozoite, which is an active, vegetative, metabolically organism. And then eventually, if conditions get bad, they can become a cyst, which is a walled-off, protected organism, which can be pretty difficult for us to kill. So as far as their life cycle is concerned, it does vary a lot depending on the species you're dealing with, especially when we're talking about the parasitic varieties. Some parasitic protozoans have very, very complex life cycles. A prime example would be malaria. I wanted to say measles, but that's fine. So malaria is a protozoan that is transmitted through mosquitoes in the equatorial regions of the globe. And it has a very, very complex life cycle. It's multiplying in the liver, in blood cells, within the mosquito itself, and it has multiple different life stages to accommodate these things. Okay? So it's not always just as simple as trophozoite cyst. Sometimes it can get a lot more complicated. And then also sometimes it is actually a lot simpler. Sometimes they're just always trophozoites and never become cysts. It really just depends on the organism and what it's trying to accomplish. Now, how do we categorize these organisms? It's usually based on locomotion or movement. Okay? How do they move? So motility by far and large is the best way to classify it. So what types of movement or locomotion can we see? Well, they can either move using pseudopods, which is basically just pushing themselves around and kind of rolling around and grabbing onto whatever they see. They can move with flagella or flagellum. This is where they have a tail that they literally wag and propel themselves forward. Or they can propel using cilia, or small little hairs that move in unison to propel the organism in a different direction. And then the one thing that's not mentioned on here is no movement at all. So there are some protozoans that do not move at all, and these are all pathogens because they don't move. So let's start off with the flagellated protists. These are protists that move using flagella. Okay? The scientific name for this group is Masticophoria. And they, again, use a single flagellum to kind of whip and propel themselves in a given direction. These often have relatively complex life cycles, and there are many parasitic species within this group. Trypanosome krusei, this causes Chagas disease. This is a disease that, once you get infected with it, causes large ulcers to form in cartilaginous tissue, primarily around the nose. And eventually, over decades, it kills you. It's primarily found in South America, and it's very treatable. However, you need to have access to health care to get treated appropriately. Giardia lamblia. So Giardia lamblia causes diarrhea. It's a diarrheal disease that is common in most parts of the world. And the organism literally suction cups to the side of your large intestine, irritating the bowels, and causes massive diarrhea. And then the last one, which you can see over here, Trichomonas vaginalis, also known as trike on the street, is a organism that targets the vagina. It's a sexually transmitted infection. It is symptomatic in females. Usually get a burning, fishy discharge, but is 100% asymptomatic in males. A lot of males are infected with this, but have no symptoms and transmit it to females. Relatively harmless, doesn't really do much damage to the vagina. However, it's a little uncomfortable. Would know from experience. Pathogenic flagellates. What your book did is they put this at the end of your chapter, or end of your slides. I brought it back up to here because it makes more sense since we just talked about it. Don't worry about flipping back and forth. This is more just for us to talk about. I'm not really going to test you on it, at least not yet. So, Trypanosome krusei, which we just talked about. This is the organism that causes Chagas disease. There's a close relative called Trypanosome brucei, which causes African sleeping sickness. Chagas disease is found in South America. Sleeping sickness is found in Africa. Okay, but they're very close relatives to each other. The way that you get infected with this organism is you get bitten by something called a retivig bug. This is also known as a bed bug. It's a relatively large bug, about this big. It likes to bite humans while you're sleeping and take a blood meal. When it engorges with blood, it fills up its guts, and so then it defecates to make some room. When it defecates the trypanosome organism on your skin, and then when you wake up and scratch the little bite, you scratch in the feces, and that's how you get infected. Once you get infected, the trypanosome, which looks like this, targets immune cells, primarily macrophages, lives within them for long periods of time, and eventually causes these open, oozing sores in cartilaginous tissue. If you don't get this treated, it eventually works its way into the lymphatic system, then the bloodstream, and you get a septic infection. However, it usually takes decades for this to happen, so it is readily treatable, but places where this is prevalent, like in certain parts of South America, healthcare can be difficult to come by, and so you still see a lot of this in certain fields. Moving on, the next group is Sarcodinia. Sarcodinia are organisms that move with pseudopods. Pseudopods, all that means is they take their cell and they literally just push themselves forward. And they grab anything in its path and it eats it. Okay? These are often called amoebas. Okay, that's their generic term. And there's two amoebas that are pathogenic to humans. Entamoeba histolytica and Nigeria flower. Entamoeba histolytica causes amoebic dysentery. This is where you ingest the organism and it literally burrows through your large intestine. And this causes ulcers and bleeding, and then you have bloody diarrhea. Nigeria floweri is even worse. This is the organism that can work its way up into your nose. It's found in fresh water, usually warm bodies of fresh water. And if you take in a lot of that water through your nose, it gets up in, literally climbs through the olfactory nerves in the nose, gets into the brain, and then just eats whatever it finds. Eventually, it will kill you in about 2 weeks. It has a 99% mortality rate, which means pretty much everyone dies when they get this. The good news is it's relatively rare. We don't see it too often. It's typically only found in young kids that are swimming and almost drowned and taking a lot of water. Okay. However, one interesting story about this is there's actually been an uptake of Neisseria flower infections, and they actually traced it back to people using neti pots with contaminated water. So neti pot, the little teapot thing you use to irrigate your sinuses, if you put dirty water in there, there's the possibility of this organism being there, and you're just pushing it right up into your nose and then into your brain. So, both of these are pretty nasty. Neisseria flower is much worse because it ultimately kills you. But Entamoeba histolytica is an important pathogen because it is pretty widespread. Okay? We actually see it a lot around the globe, not so much in the US, but elsewhere. And it causes something called amoebic dysentery. In this case, what you would do is you would ingest a cyst. These would usually be found in food or water, but it's most problematic in water. The reason for that is the cyst is very, very hardy, and so chlorine will not kill it. So the chlorine in our water does not get rid of it. The only way to get rid of this guy is to mechanically filter it out. Once you ingest the cyst, the stomach acid opens it up. It becomes a mature protozoan at that stage, and then it works its way down into the large intestine. It then burrows through the large intestine and can actually get into the bloodstream and even the liver in some cases. If you have a high number of these organisms in your gut, it will perforate the colon and the large intestine to the point where it becomes what we call Swiss cheese colon, and it needs to be resected or removed. Otherwise it will die. But the good news is that this is typically not that big of a problem unless A, sanitation is really bad, and B, you are immunocompromised. So if you have a healthy immune system, chances are this will not kill you or cause any serious problems. If you're immunocompromised, however, it can be a problematic infection. So the life cycle, as we just talked about, you ingest the egg wakes up, matures into the trophozoite, and works its way into the large intestine. Wherever the adult is found, and this goes with any parasite, wherever the adult is found and wherever it can mate, that is considered the final or definitive host. So in this case, it likes to be in humans, it mates in humans, so this would make humans the definitive host for this organism. The next group, Ciliophora. Ciliophora use cilium. Okay, it's a movement. There is only one pathogenic variety of ciliated protists, and that is Blantium coli. It's a relatively rare pathogen, doesn't cause much problems other than some basic mild diarrhea. Okay, so not that big of a deal. The next group, however, the Apicocomplexa, these are organisms that lack movement altogether. Okay, they do not have any way of moving around on their own. So instead, that means they have to be parasites because they use our body to move around and get from point A to point B. There's many important pathogens in this group. Two of the biggest, Plasmodium vivax and Toxoplasma. Plasmodium vivax is one of the organisms that causes malaria. This, again, is an equatorial disease. It's transmitted through Anopheles mosquitoes. And it basically reproduces in the liver and the blood cells, eventually killing blood cells and causing anemia and potential death. Toxoplasma gondii is a protozoan found in kitties. All cats have this. It's a normal commensal found in their gut. And we can pick it up from contaminated products from the cat. I'm not saying you have to eat the cat shit or anything. It's just a very sticky cell, which gets all over the place. And I can almost guarantee you, if you have a cat, you've been exposed to this. It doesn't really do anything to humans unless you're immunocompromised or you're pregnant. If you're immunocompromised, it works its way up into the brain and can cause problems like neurological issues. If you're pregnant, it can cross the placenta, targeting the fetus and causing problems. Yes?
That's the toxoplasmosis, right?
Yes.
Okay.
So toxoplasmosis is caused by Toxoplasma gondii. And that's why when you're pregnant and you have cats, they tell you to either wear gloves or get someone else to clean you. Just Just be careful because cats are gross. At least mine was. He liked to sleep on my pillow, on my face, and whatever. So it's hard to get away from it sometimes. But the good news is that we don't really see too many of these in the U.S. because it is treatable as long as you catch it early. All right, so how do we identify protozoans? Well, typically we can look under the microscope and that tells us a lot. Because we can look under the microscope and determine its locomotion and all of these other things. The way that we would do this is we would collect a specimen from wherever the patient is infected, and then we would look and see what we find. Okay? If you have a protozoan that lacks locomotion in the blood, it's probably malaria. Right? So you can kind of deduce what it is just based on where it is and what it has or what it does not have. However, there are other ways of determining what type of protozoan you have. One of the easiest is using serology, which is where we look at antibodies and our immune response to that protozoan, and that will tell us what we potentially have. As I mentioned, there are a lot of protozoan pathogens. We will talk about these in more detail later on. We've already talked about a few of them. So last but not least, what we're going to do is talk about the helminths. Helminths are multicellular eukaryotes. Okay? These are often large. They can be meters in length in some cases. So strictly speaking, they're not just microorganisms. Okay? However, the eggs that they produce are very tiny, requiring a microscope, and so this is why they fit into our realm. We usually break them up into two groups, the nematodes, or the roundworms, and the platyhelminthes, or the flatworms. Okay? So roundworms and flatworms. And within the flatworms, you have tapeworms, which are segmented, and you have flukes, which are non-segmented. So flatworms, these are flat, hence their name. They are thin, and flat, and they are in two subdivisions, cestodes, which are segmented, and trematodes, which are not. Here is an example of a cestode. This is a common human tapeworm. This guy can get up to 2 meters in length. It doesn't really do any damage to a human. Instead, it just attaches to your small intestine and takes in some nutrients every now and then. Okay. The way it reproduces is it has these little segments. These are called proglottids. And in these proglottids, you have a male and female reproductive tract. These will break off every week or so, reproduce, and then release eggs in their feces. Over here, we have a trematode. This is a liver fluke. It's relatively large. You can see here's 1 millimeter, so about the size of a centimeter, centimeter and a half. Looks kind of like a flattened slug. And these guys like to go into places like the liver or the lung, and they sit there and they sop up some nutrients from the blood in the area. Like most parasites, especially with the helminths, as long as you don't have a very large number of these organisms in your body, they're not going to do much damage. Most of the time you have them and don't even realize it. In fact, there's an argument out there that having parasites is almost a good thing. The more parasites you have or exposed to in your life, the less allergies you tend to have and problems with the immune system. Roundworms, also known as nematodes. These are elongated, cylindrical, or round, and they are unsegmented. One example of a roundworm is a pinworm. I'd like to talk about pinworm because this is one example of a helminth that is common in the United States. Most helminths aren't very common in the U.S. because we're very clean. And a lot of these get transmitted through fecal-oral contamination. So just being clean is enough to get rid of them. But pinworms, however, are still found a lot in the United States, and that's because they primarily target young children. And young children aren't that clean by nature. So pinworm, the way that it works is you ingest the eggs. The eggs become larvae in the stomach. They then migrate into the small and large intestine, becoming adults. And once they're adults, they then mate very regularly. Once they mate, the female will migrate out of the anus while you're sleeping and lay eggs around the ring of the anus. These eggs are sticky. They're also immunogenic, which means when you wake up, they cause itching. You wake up, you scratch your butt, you get the eggs stuck on your hands, you then go eat or suck your thumb or touch your friend, and you can transmit the eggs that way. Just like most helminths, you can auto-infect yourself very easily, which means you just keep infecting yourself with more and more eggs. So in this case, every time the kid wakes up in the morning, scratches its butt, sucks its thumb, it's getting infected with more and more and more worms. Until it gets to the point where you start pooping out worms because there's so many of them. And that's usually how parents find out about this. Another way of determining whether or not your kid has this is the Scotch tape test. And this is where you take double-sticked Scotch tape and stick it on their anus. It's gross, but if you get little eggs that get stuck to it, that tells you that your kid has completely harmless, does nothing to the child, doesn't take away, anything. However, it's just a little gross. It's also very difficult to get rid of because these eggs are very hardy and sticky. So here is an example. You have a kid scratching their butt and sucking their thumb at the same time. And like I said, this is more common in kids because, when you're an adult, you scratch your butt and you're like, oh, I should probably wash my hands, right? When you're a kid, you don't think about that kind of stuff. And so it's very easy to spread. Also, when you're a kid and you're in a daycare setting, you've got a bunch of kids scratching their butt, it's very easily spread throughout that population. They said it's difficult to eradicate because these eggs are very sticky and hard to kill, but it is not life-threatening, it's not a serious thing. It's kind of like getting lice. Right? So how do we classify helminths and identify them? Okay? So we classify helminths based on their appearance. So are they segmented, are they round, are they flat, all those kinds of things. And we also can identify them based on their appearance. Usually it's the appearance of their eggs. The way that these guys reproduce, a lot of times is they'll release eggs either in the feces or the urine. So if you get some feces and you chew it up and then you look under the microscope and you see a bunch of eggs, that tells you the person has a helminth. And a lot of times these eggs can be very distinct. So if it has a little pointy thing here or a weird dot here, that tells you it's this. Right? So we can use that to come up with a positive indication. Also, we can look at things like serology. So again, what type of antibodies do you have, and that will tell you what you've been exposed to. How common are helminths? Very common, at least on the worldwide scale. They are by far the most common type of infection. There are literally billions of people that are infected with helminths around the globe. Okay? Very, very common. And as I said, most of these people live normal, healthy lives. Helminths, as long as you don't have a lot of them, and this is just generally speaking, there are some exceptions, are not that harmful. They live in you, they have evolved with us so that they can just hang out there, take a little bit of nutrients, and then do their own thing. Okay? It's when you get a lot of these, or you get infected with a helminth that wants to go into another animal, like a pig or something, that's when it can become problematic. But traditional human helminths, for the most part, are not that big of a problem. So here's some examples. We'll talk more about these later on in the semester. So here, Ascaris is another common, it's also known as roundworm, very common helminth in humans. But then there are some, like pork tapeworm, for example, that are typically found in pork, but if we ingest them, they tend to get a little confused because we're not a pig, and then they start going around in places they shouldn't, and that's when they can be problematic. Okay? So I don't want you to think that helminths are a good thing, but they're not necessarily the end of the world. All right, that's it for chapter 5. Any questions? All right, let's go on upstairs.